This is a production of Cornell University.
So today we'll be talking about the Malus-Venturia Pathosystem and how host adaptation and geographical isolation leads to population differentiation and the impact that it has on host resistance breeding in apples. So the fungus Venturia inaequalis is an ascomycete fungus. That's the causal organism of apple scab. It causes olive green to black lesions on the leaves and these black cracked appearances on apples. This is a very significant disease in apples. Producers have to undergo really extensive management practices in order to control it because these cracks severely reduce the fruit quality and can just completely decimate producer yield. The spores have a very limited dispersal area, but they do undergo a sexual phase in their yearly life cycle, and frequent recombinations lead to high variability within the fungus. So for management of apple scab, like I said, it's a really significant disease, and primarily management focuses on proper orchard sanitation, so making sure that leaf litter is removed at the end of the season because that is where the sexual recombination occurs for the fungus. So proper removal of leaves and destruction of that inoculum is really important. Careful fungicide application to make sure you're not leading to fungicide resistant populations. And then also developing resistant cultivars is really important for stable long-term management, especially for organic growers who don't necessarily have the same chemical tools that conventional growers do. So in apples, there are currently 18 identified major genes that are resistant to apple scab. We call these RVI. And there's a gene-for-gene interaction between these Malus RVI genes with pathogen AVR genes. And these effector proteins trigger the downstream immune response. And about 9% of scab-resistant cultivars rely on a single gene, RVI6. This is originally identified in Malus floribunda 821. This resistance has since been broken. It's been repeatedly identified in Europe, but last year it was actually identified here in New York as the first case in North America. This is really a cause for concern. That RVI gene is in a tight gene cluster on Lucas group 1. And another one of my projects is looking at another candidate host resistance gene from Honeycrisp that's also located in this tight gene cluster between RVI6 and RVI17. So apple domestication originally started in the Tianshan Mountains in what's now Kazakhstan. And that's the center of origin for the primary progenitor species for apples, Malus-Venturia. Malus-Venturia was brought along the Silk Road trading route where it hybridized with other apple species in the area, primarily Malus orientalis in the Caucasus area and Malus sylvestris in Europe. And this hybridization was really important for the coevolution of Venturia inaequalis because the host geographical movement towards Europe, as well as the hybridization with other local species, led to 3 really identifiable populations of the eye. So there's the Central Asian mountain population that's only found in the Tianshan origin of Malus-Venturia. There's the Central Asian plains population, which can infect Malus-Venturia and Malus domestica, which are our moderate domesticated apples. And then there's also an EU population that only is identifiable in Europe, and these populations can overcome Malus sylvestris and Malus domestica. So these 3 populations are really clearly identified and well reported on. But one of the things that we are really interested in is looking to see if the host differentiation and geographic isolation of North American isolates led to a new unique population. So we're looking at the geographical isolation between Europe and the United States, as well as any host adaptation that might have occurred with North American isolates. So we started out with 164 isolates that we collected both from NCBI, the publicly available, and from our lab. We have Illumina short read sequences from them. We removed 41 of the isolates, either due to poor sample quality or contamination determined by the mating type locus analysis. And so we aligned these sequences against the NCBI reference genome. We filtered out the alignment reads for quality, and then we conducted SNP calling with the GAPK haplotype caller. So we ended up with a little over 19 million raw SNPs, and we ended up with SNP filtration over a couple of stages to make sure that we weren't losing out quality SNPs as we were going through these bulk reductions. So we started off with just some very basic filter parameters to try and remove the lowest quality sites. That brought us down to about 4.4 million. And then we wanted to look at population level SNPs to bring that data set down to a workable range. So that brought us down to 375,000. And then finally, we did hard heterozygosity filtering because Venturia is a haploid fungus. And so any heterozygous sites would just be due to sequencing error. So principal component analysis appears to show our North American isolates starting to form their own distinct subpopulation. You can clearly see other populations identified that are due to both host geographic adaptation and geographic isolation and host adaptation. You can see the European Malus, and it's covered up by the zoom bar up there, and then the two other identified populations, the cam and cat populations, those are the Central Asian Plain and Central Asian Mountains, as well as the population that was obtained on Malus orientalis. So PCI appears to show a population developing. However, further Bayesian analysis shows that there's not enough statistical support to show that these isolates have formed their own true population yet. What it likely means is that there just hasn't been enough time for that population to develop. Apple domestication took place over a range of thousands of years, but these isolates have only been in North America for the last few hundred. So we really just haven't seen these isolates form their own distinct population yet. And also, it was the process of both the host adaptation due to hybridization and the geographical movement that led to these distinct categories. So I think that also played a role in why we haven't seen a true population arise yet. But looking at just isolates that we collected in Geneva, New York, up at Cornell Agritech, we actually did identify 3 subpopulations within a single orchard. And the key characteristic that defines these subpopulations is the ability to overcome that key resistance gene, RBI6. So 2 of the populations have the ability to overcome, but there are 2 very distinct populations that have that characteristic. So what that likely means is that either 2 resistance overcoming events happened in the same orchard, or an isolate that can overcome was introduced later on. And this orchard did not have any kind of disease management, so that probably also is very significant for allowing that selection pressure to mount. So just kind of wrapping up, these North American isolates haven't yet formed a distinct population just due to the limited time that they've had in isolation from other key populations. And this work in the future can help identify those pathogen ADR genes, which are really important for identifying new populations of the pathogen that can overcome host resistance as breeders continue to develop new resistant cultivars. And also identifying the SNPs that occur in these effectors can also help us try and understand how the host adaptation and the geographic isolation actually led to these population differentiation and how this might happen again in the future. I'd like to thank the Khan Lab for their support, as well as the other 2 members of my committee, Sarah Petherbridge and Lance Cadle-Davidson.
I have a question.
Yes.
And how long do you think will isolation of new cultivars take in like a time frame? Like how long do you think it would take for a like US or North American population to form, or like even to spread through the US with like a time scale?
Not 100% sure, and part of the reason why that might not be the case is just due to how freely isolates are able to move between areas now, just due to global trade. So originally, that limited spore dispersal played a key role in why the isolates were able to slowly evolve with the co-domestication process. But now isolates can travel between Europe and North America in a few hours, much along with infected materials. So rapidly being able to go back and forth might mean that they might not ever form their own unique population.
Yeah, that makes sense. Can you transfer the disease with, if you bring like the apple fruit, an infected apple fruit from Europe to the US, for example, or does it have to be leaf material?
No, you can bring infected fruits as well. But typically infected fruit isn't transferred as often. The blemishes that apple scab causes are so devastating to the fruit price that most farmers don't even bother trying to harvest them if they identify them in the field.
Thank you.
Yes.
So do you have enough understanding of the effectors and the translation to be able to start structurally modeling it, you know, at the level of protein-protein interactions?
Not yet. Part of that is that Venturia effectors are still a very emerging area of study. We just received a set of candidate effector sequences from Carl Meserich in New Zealand. And we have annotation data for these SNPs in this reference genome. So part of what we're going to try and do next is look at the SNPs within these candidate effector regions and look at the predicted effects they have on the protein translation. And hopefully then we might have a little bit better understanding, but not quite there yet.
So you think they affect rate of translation or the actual protein structure?
The protein structure.
Exactly. It was kind of, you know, just, you know, where we are now with, you know, fold proteins predict their structure. You know, this whole question of being able to dock things you don't have crystallized now,
Yeah.
I think really opens up and maybe.
Oh, thank you, Ian. And let's applause to everyone who spoke today.
This has been a production of Cornell University, on the web at cornell.edu.